So before this video starts, you're going to want to have these files downloaded. These are the locations. You're going to want to have PyCharm downloaded, and you should be good from there. We're going to go ahead and set the target and go and find it.
Okay, so I gotta rush this video out here. I wanted to get it out about last week with the lecture 1 to follow up that coding, because this is where I left off, except I had some weird names in and I kind of corrected it to make sure that people can follow along. So the first one is the NCBI file, and the other one is going to be the GSI AID file. With the UK1.1.7 variant, which is a two amino acid deletion, and I linked an article about it in the information, so we'll go over that here in a moment. I just want to explain what's going on the screen right now, and that's that well, we have some checks here to make sure we're on the right file that I showed in the video, and I changed it up to the first codon here, and basically what this is going to do, it's going to find the first time that this repeats. Okay, enough about over here. Real quick, down here we know that this sequence right here, it repeats at this number in the file, and then the count, this right here, this 9 segment, only happens one time in the file. So that's why this little area is important, because with that information we can go ahead and set up a nice couple of variables here. So in the video I did a calculation for the basically I just took the sum of those numbers that were listed in the NCBI for the gene, and then applied them to the start codon, and then I basically made something so that I could easily display some info here. So we have these happening at these locations right here. So we have our this segment happens at 21648 in the sequence in the file, and then this one happens at that number. So we've now set these two variables to mean those two numbers, and we can then basically identify GeneS and print it out. So let's go ahead and see what that looks like. So we're setting right here, we're setting GeneS to our reference sequence, and then basically going between these two areas right here, StartGene and StopGene, and these are the two variables that we set up that basically equate to the S gene. And I'm going to go ahead and print that out here. Let's see what it looks like. And what you could do is you could match this up with the NCBI information, the last couple of base pairs. I mean, if they match, you can see that 9 of them happening in 30,000 base pairs. We only have 1, so that's a pretty good indicator that you're in the right spot. Okay, so let's see here. Let's move on to the next. Okay, so this is all fine and dandy, but it's still just listed out, and we really want to represent it like you'd see it being built. And the amino acids, basically every single set of 3 of these are going to be an amino acid, and what we're going to do here is we're going to write an iterating statement. Now I have another video where I kind of teach iterating statements a lot better, and basically here it is. Here's the statement. So we're gonna go G, and so basically what this is gonna do is every 3 spots, it's going to separate the numbers. And what it'll look like int s, and it'll look like this. So this is a nicer view. We have our amino acids in sets 3. So now we can hide these, all the base pairs, and go to the and now what we really want to do is we want to identify these in an easy way. So we can easily make an amino acid library by taking the so you have your NCBI here, and what I'm going to do is I'm going to go up to the S gene. Okay, here's the S gene, and if you copy this, you can actually make yourself an amino acid library pretty easy for this gene, and that's what we're going to do here. Except I already have it made, so when you go to copy and paste it right from there, you're going to have a lot of spaces. I mean, I went through manually and deleted all the spaces, so I didn't have to make y'all watch that. And then I built another iterating statement that says every single letter, you see this is every third letter, it's going to make a space in the file. This one is every letter is going to be spaced in the file and made to its own spot. So what it's going to look like when you have them stacked on each other is print AAS. I just noticed that. Okay, hilarious. And there we go. So we have an amino acid library that's going to line up over our genes, or not genes, our amino acid base pairs, and we'll get a better understanding with this. Okay, let's refocus here. Calling a refocus, and let's throw up the target. The target is the spike H69V70 deletion. And how do we know that we got the right spot? Let's go ahead and put it on the screen. Now I made a little change here in the code, a little correction, and now I'm going to throw a little print up. So we're going to print spot 68 and spot 69. So the reason why we're going to do 68 and 69 is because in computer language they don't start with 1 like we did in our numbering here for the or like we do in this numbering for the H69V70. The first amino acid is listed as number 1, but in computer code they start with 0. So what you could do here is you could put like an X or anything to make it 1 bigger, and it'll line right up with 69 and 70 if that's what you need. But in this channel we understand computer code just fine, and that it starts with 0, so we're going to start 1 back. Okay, so then what we're going to do is we're going to make another file, another code for the B.1.1.7 strain. And let's see here. I'll have to do a video on nomenclature to understand how they name them. It's really just every time a different set of variants show up, they extend it if it had previous variants. And let's see here. Okay, so I stopped and edited, and this is what I kind of cleaned up right here. And so it's StartUkGene, StopUKGene, and what we're going to do here is we're going to erase this little print area and copy this gene S, and we're going to make a UK form, or the UK variant, ukGeneS, and what did we name it? We named it UKrefseq. And start. Stop. And there we go. So now we have to do our iterating statement to break it up, so it'll just be one big line. But and let's just always make sure that we got it working. So let's do a print UK. UK. There we go. Well, this is it right here. Okay, so I got a little lost there because the ukGeneS area, that was not reading properly, and then I had the font just way too small to be figuring out problems. So what I'm going to show here is that we're off. This is not the right location, and I'm going to show how I'm going to figure it out, what is going wrong. And the first thing I want to do is just clean up this little area right here. I don't need this. To figure that out, I'm going to put all the prints. I'm going to hide those too. I don't care for those right now. Let's see. Okay, so here we go. We have our first codon here. No, this is what we got. Okay, so we want, oh, that's what happened. So in my haste, I did not drop down the right file here. Right here. So I kept the reference sequence, and I needed to bring down the UK reference sequence. So I missed that, and then I caught it because I reprinted that S. I mean, it's a good thing I didn't just go right through it. So that's why you always want to put little checks in your code to make sure you're in the right locations. Now that we've corrected that, we can continue where we left off, and it was right here we were making the UK file that is going to be all split up into 3s. So let's go ahead and make that. It's going to be UKS and then equals, and basically we're just going to copy this right here, run the same iterating statement, and replace it with the ukGeneS, and every 3 it's gonna chop this UKS up too. We're not going to do an amino acid on this one, just because we'll have it overlay, and then you can delete the deletions. Now we're going to do some basic prints here. So I'm going to print out spot 68 in the amino acid sequence. So these the amino acids that are coded, and then 68 in these other slots too. So you can see that at slot 68 and slot 69, or position 69 and 68 in these files, they're not the same. So we do have our histidine and valine here. These are the deletions, the 2 amino acids that get deleted in the spike protein. And so our reference file is showing histidine and valine, but it's also showing 2 different other amino acids. So, and that reason is that it deleted and then it shifted over into the positions. So if we wanted to look a little upstream, we'd go to 66. So let's look at the amino acids at 66 and 67. So now they're overlapping more here. And what we're really seeing here is that 66 there was no deletion, it looked good. And then at 67 there was actually the beginning of this deletion. The thing is that ATA and both ATC both code isoleucine, so it made it out with just a two amino acid deletion, and it deleted some other stuff too here. So let's go ahead and take a look at that. Oof. We're ready now to display the whole, or a whole little set of sequence. So what I'm going to do here is I'm going to do the amino acids on top for 65 to position 72 here. So I'm going to go ahead and display that. We have our amino acids that are getting coded. We have isoleucine here, and this is the beginning of our deletion. We have ATA and ATC. Now both these code isoleucine, and you can see that this ATCT right here, GT, these 5 base pairs also get deleted. This CTCTGG gets then shifted over to the left here in this bottom one, and that's the B.1.1.7 strain of the coronavirus. So we have this deletion happening. Isoleucine is still being coded. We are then losing histidine and valine, and then it just continues right on. So we're losing histidine, which has a polar, my bad. We're losing histidine, which has a charged polar side chain, and then we're losing valine, which has a non-polar side chain. We're ready now to display the whole, or a whole little set of sequence. So what I'm going to do here is I'm going to do the amino acids on top for 65 to position 72 here. So I'm going to go ahead and display that. We have our amino acids that are getting coded. We have isoleucine here, and this is the beginning of our deletion. We have ATA and ATC. Now both these code isoleucine, and you can see that this ATCT right here, GT, these 5 base pairs also get deleted. This CTCTGG gets then shifted over to the left here in this bottom one, and that's the B.1.1.7 strain of the coronavirus. So we have this deletion happening. Isoleucine is still being coded. We are then losing histidine and valine, and then it just continues right on. So we're losing histidine, which has a polar, my bad. We're losing histidine, which has a charged polar side chain, and then we're losing valine, which has a non-polar side chain. Cleaning this up here, let's just re-emphasize before we change it to just reinforce what we're looking at here. So what we're looking at here again, just to emphasize, there was a deletion that happened right here, and it shifted over, and then we have GG, ACC, AAT, and that it's going to continue for so on, except it lost that little segment that coded the H and the V. So let's go over to this left hand side over here where it says the GIS AID file, and I do apologize, I said that wrong in the beginning of this video, and they did a great job, minus the only giving access to people with collegiate email addresses. They're doing a great job. So let's go ahead and let's search these missing, or not the missing ones, but this little segment right here. So CTCT GGG ACC, and look, we already got that one spot where it could be. We know that this C is still coding that isoleucine, and then now let's put in 6 X's for that deletion. There's 6 X's, and let's go ahead and rerun that, and now we have a nicer view. We see in by deletion with these X's, and it continues on. So that's how you can do it.